Good morning, dear students. Today we are going to discuss lesson 3, domain bacteria. This is a typical bacterial cell. It contains flagella, nucleoid, cell membrane, cell wall, capsule, ribosome, pilus. This is smaller hair-like structure, pilus, and this is longer hair-like structure, flagellum. Now I will discuss the important features of domain bacteria. These are prokaryotic organisms, no true nucleus, and usually they are unicellular, and their DNA located in the nucleoid, and their cell wall contains peptidoglycan, and they usually have a single circular chromosome. Now we will discuss bacterial genetic material. Chromosome, usually a single circular DNA molecule in the nucleoid. Plasmid, these are smaller circular DNA molecules separate from main chromosome. This is main chromosome. Plasmids can be transferred between bacteria. They may contribute to genetic variation and adaptation. This is biggest chromosome, nucleoid chromosome, main chromosome, and these are smaller circular chromosomes known as plasmids. Bacteria come in contact and form a tube, a channel through which plasmid can pass from one bacterium to another bacterium. That is donor bacterium, and that is recipient bacterium. Reproduction in bacteria, it is generally divided by asexually by the process of binary fission. One bacterial cell is divided into two. It's known as binary fission. Genetic material replicates and cross wall starts to form. Eventually parent cell is divided into two daughter bacterial cells. Bacteria primarily reproduce asexually through binary fission. A single bacterial cell divides into two identical daughter cells. Binary fission allows rapid population growth under favorable conditions. Nutritional methods in bacteria. Autotrophs, they are self-feeding organisms that make their own food. Example is photosynthetic bacteria. Heterotrophs obtain food from organic matter. Example, decomposer bacteria. Morphology of bacteria, forms of bacteria. Cocci, these are spherical shaped bacteria. Bacilli, these are rod shaped bacteria. Spirilla, these are spiral shaped bacteria. Vibrios, these are comma shaped bacteria. Cocci bacteria, these are cocci bacteria. You can see these are spherical. Bacilli bacteria, rod-shaped bacteria. You can see the diagrams. Spirilla, these are spiral-shaped bacteria. Vibrios, these are comma-shaped bacteria. Arrangement of bacterial cells, how bacterial cells arrange themselves. Bacterial cells may occur singly or in characteristics arrangement. Diplococci, cells arranged in pairs. Streptococci, cells arranged in chains. Staphylococci, cells arranged in clusters. Other arrangements may occur according to species specific characteristics. Arrangement of bacteria, you can see Diplococci, cells arranged in pairs. Streptococci, cells arranged in chains. You can see bacteria are arranged in the form of a chain. Staphylococci, cells are arranged in clusters like this. This is a cluster of bacterial cells. Other arrangements may occur according to specific characteristics. Tetrad, Sarcini, palisade, groups of 4, tetrads, group of 4, Sarcini a packet, palisade side by side. These are some other arrangements may occur according to species specific characteristics. Flagella, that is an outgrowth for locomotion, movement. Many bacteria have one or more flagella. Flagella are whip-like structures. Their main function is to enable bacterial movement. This is a bacterial cell and this is flagella. It plays a role in movement. There is different arrangement of flagellas on bacteria. This is a pattern on both sides, on whole surface. This is the structure of flagella that plays important role in the movement of flagella that results in the movement of bacteria. Pili and fimbri. Pili and fimbri are hair-like structures found in some bacteria. They help bacteria attached to surfaces. Some pili also help in exchange of genetic material between bacteria. These are pili a little bit longer, and fimbri, these are smaller, shorter, but more in number. Fimbri and some pili enable bacteria to attach to both living and non-living surfaces. Bacteria is attached to the surface with the help of fimbri and some pili. By these fimbri and pili, bacteria can attach to tooth surfaces, attach to plant surfaces, attach to medical devices. Exchange of genetic material. Sex conjugative pili form a bridge between bacteria and help transfer DNA plasmids. You can see pili come in contact, a tube is formed, and through this tube plasmids can be transferred from one bacterium to another bacterium, and you can see here. Respiration in bacteria, obligate aerobes. These types of bacteria require oxygen. Obligate anaerobes. These bacteria cannot tolerate oxygen. Facultative anaerobes. They can grow with or without oxygen. Microaerophiles. They require low concentrations of oxygen. Aerotolerant anaerobes. They tolerate oxygen but do not use it for respiration. Some bacteria use fermentation to produce energy without oxygen. Some bacteria can survive and grow in extreme environmental conditions. The thermophiles, they can thrive at high temperatures. Halophiles, they can thrive in environments with high salinity. Acidophiles, they can thrive in environments with low pH. Pathogenicity. Some bacteria cause diseases in humans, animals, and plants. Pathogenic bacteria may produce toxins. They may also produce other virulence factors that help them cause diseases. Symbiosis. Many bacteria live in symbiotic relationships with other organisms. Bacteria in legume root nodules, Rhizobium bacteria fix atmospheric nitrogen for the plant. The plant provides food and shelter for bacteria. Gut bacteria. Gut bacteria help in digestion, synthesize vitamins, and protect from pathogens. They get food and a safe habitat. Commensalism. One organism benefits while the other is not harmed. Bacteria on human skin. Bacteria live on the skin, obtaining food and shelter. The human is not affected. Symbiotic relationship between bacteria in the mouth. Bacteria benefit from food particles and habitat, and host is unaffected. Symbiotic relationship help maintain balance in ecosystem and are important for the survival of many organisms. Major groups of bacteria: Proteobacteria. Proteobacteria, diverse group, include many pathogens and beneficial nitrogen fixing bacteria. Firmicutes, gram-positive, many are spore forming, include helpful and harmful species. Actinobacteria, gram-positive, high G plus C content, include filamentous forms, important decomposers. Cyanobacteria, photosynthetic, oxygenic, important in aquatic and terrestrial ecosystem. Spirochetes, spiral-shaped, motile, and endoflagella. Many are pathogenic. Acidobacteria, found in acidic soils and environments, diverse metabolic capabilities. Acidobacteria found in acidic soils and environments, diverse metabolic capabilities. Aquifici, thermophilic, found in hot springs and deep sea hydrothermal vents. Now we will compare archaea versus bacteria. Archaea, ether linkage is present in cell wall. Bacteria, ester linkage is present in cell wall. And branched chains of polypeptides and generally unbranched chains in cell wall. Branched chains in cell wall. Generally unbranched chains in cell wall. No peptidoglycan. Peptidoglycan is present. Methanogenesis in some bacteria. Methanogenesis in some organisms. No methanogenesis in bacteria. Similarity, both are prokaryotic and usually have circular chromosomes. Key terms: variety of life, that's biodiversity. Prokaryote, cell without true nucleus. Eukaryote, cell with a true nucleus. Phylogeny, evolutionary relationship. Methanogenesis, production of methane. Peptidoglycan, major bacterial cell wall component. Plasmid, small circular DNA molecule. This is all about the topic domain bacteria.